and the collecting duct, which is what I still had to talk about last night with the Tuesday-Thursday session. So tonight we're going to look at the structure of the bladder, all right? A little bit of the micturition reflex, which we very similar to what we talked about with the reproductive system as far as sensory and sacral-splanchnics and pelvic-splanchnics, those same structures. But I want to review with you how sympathetic and parasympathetic control micturition or avoiding. And then we're jumping to the acid-base balance. Okay? We got a quiz in there too. So whatever time you need to study for the quiz, we'll do kind of as a break in between. So we have the filtrate, remember we still call it filtrate, coming through the papillary ducts into the minor calyx and major calyx and flowing into the pelvis. And then the pelvis narrows to become the ureter. So the ureter is still traveling behind the peritoneum, and it's passing into the posterior aspect of the urinary bladder. So let me get a urinary bladder muscle up here. And the openings for the ureters run at an angle that forms a natural valve. So as the bladder fills and stretches out, that functions to close off the ureter so it doesn't overstretch. All right. So let me get these structures labeled. What's the lining epithelium of the ureter? Transitional epithelia. Okay. And the lining of the bladder. So everything from the minor calyx down to the, I'm going to make this a little bit more narrow, down to the urethra, except for the very tip of the urethra is going to be transitional epithelium. All right. The smooth muscle of the urinary bladder has a name. We don't often name smooth muscle. We call it smooth muscle in the wall of the scrotum. Anybody remember the name of that? Started with a D. Dartos muscle. The skeletal muscle surrounding the spermatic cord had the cremaster name, but that's skeletal muscle. We named the trachealis for the posterior wall of the trachea. And the smooth muscle of the bladder is known as the cremaster, detrusor. Now we have another smooth muscle component of the bladder, which is an internal sphincter. So that's also smooth muscle, and it's identified as the internal urethral sphincter. We'll see commonalities to this when we get to the digestive system. And then the urethra continues. Now, if this was the male, we would have the external urethral sphincter down here because we would have the prostate gland in between. In the female, it's right underneath the internal urethral sphincter. So we'll make this a blend between the two. Actually, we'll make this a little bit shorter and put in the external urethral sphincter. This is skeletal muscle. It was also our external, our deep transverse perineal muscle, if you recall, or the urogenital diaphragm. Okay, skeletal muscle. Just remind us of why I drew it in different colors. And then we'll continue the rest of this as the urethra. Most of the urethra, especially in the male, is going to continue to be transitional epithelia. But the last centimeter or so is back to non-cornified stratified squamous. What we see lining the mouth, lining the last couple inches of the anal canal, and so on. Okay? So for the female here, it would be about this much. Would still be the non-cornified stratified squamous, but everything else is going to be transitional. Yes?
Oh, no, it's just...
Oh, okay, nothing yet. So the mucosal lining of the bladder is thrown into folds called rugae. Remember we used that term for the vagina? And we'll see it again in the stomach. That just allows expansion and folding as the urinary bladder empties. So these areas of mucosal fold are referred to as rugae. But in this area between the two ureteral openings and the urethra, this triangular area is without fold. It's still transitional, but it's without fold. And that triangle area without rugae is known as the trigone. Okay? So right here. All right, so that's the anatomical components. Now let's look at the physiological aspect of its control. And would the, remember whether it's sympathetic or parasympathetic, that function to cause the bladder to contract? So going to the bathroom, I guess is what I'm asking. Would that be sympathetic or parasympathetic?
Parasympathetic.
Parasympathetic. So as the bladder is filling, parasympathetic is going to be inhibited and sympathetic is going to be active, all right? So the bladder filling or holding urine is going to be a sympathetic dominant situation. So we're going to be emphasizing the sympathetic effect, which is inhibition of contraction of the detrusor muscle and constriction of the internal urethral sphincter. So here again, we have the autonomic nervous system. We have norepinephrine causing relaxation of this muscle and contraction of this muscle. And the reason, even though it's the same neurotransmitter, is what's inside the cell. It's one of the enzymes inside the cell, all right? So this is going to be under sympathetic influence. And it's going to result in detrusor muscle relaxation. And internal urethral sphincter contraction. All right, and what's the external urethral sphincter going to be doing? Contracting or relaxing? If we're filling the bladder and holding the urine, it's going to be contracting as well. So these are going to be postganglionic sympathetic neurons releasing norepinephrine and being inhibitory to the detrusor muscle, all right? And postganglionic sympathetic neurons being active to the internal urethral sphincter to cause it to contract. And what nerve is our motor neuron to the external urethral sphincter going to be found in? Lower motor neuron in the sacral spinal cord. What nerve is this going to travel in? What's the nerve to our bulbospongiosus muscle and our ischiocavernous muscle?
The pudendal?
Pudendal nerve. Yeah, so this is going to be a lower motor neuron in the pudendal nerve. And it's going to be active as well with contraction of the external urethral sphincter. Okay? So all of that is the sympathetic allowing the bladder to fill. Then as the bladder fills, it's going to be stretching the detrusor muscle. Okay? And that's going to set up, that's going to affect the activation of stretch sensory components. Those are also going to travel in the pudendal nerve. All right, since a neuron for stretch is going to travel in the pudendal nerve. So if I fill up our bladder with urine here, I don't know why it's orange, it just is. Okay, so we're going to be sending stretch receptors to the spinal cord via the pudendal nerve. And now those are going to inhibit sympathetic and activate parasympathetic. Now this diagram involves the micturition center in the pons, and we're not going to worry about that. Okay? So I just want you to recall that our sympathetic is coming via what nerves from the lumbar region to get to the bladder? Splanchnic, sacral splanchnic. Okay? So these are going to be sacral splanchnic nerves. As the bladder fills and sends sensory input back to the spinal cord, that's going to inhibit the sacral splanchnic and activate the parasympathetic system, which is pelvic splanchnic. All right? So I know it's an overlap here of colors and everything. Let me use black for that one. So you can refer back to these. But we're going to have then our preganglionic parasympathetic coming to our postganglionic sympathetic. Preganglionic parasympathetic coming to the bladder wall, which contains the postganglionic parasympathetic. So this is acetylcholine. All right, so I'll kind of type in there. I'll put a legend over here. So this is parasympathetic, pre and postganglionic. And so that nerve would be the pelvic splanchnic, forming the preganglionic. So what's it going to do? Plus or minus here for the detrusor muscle? Because now we're full and it's time to go to the bathroom. A plus. So this is going to result in contraction of the detrusor muscle. All right? So what do we need to do to our internal urethral sphincter? It needs to relax. Okay? So acetylcholine in the detrusor muscle is going to cause it to contract. Acetylcholine in the internal urethral sphincter is going to cause it to relax. So if you're a baby or a toddler or an adult with spinal cord damage and you don't have use of those autonomic neurons, then as the bladder fills, it's going to contract the detrusor and relax the internal urethral sphincter, and you're going to go to the bathroom. Okay? However, starting anywhere from 1 to 4 years of age, we learn to control our external urethral sphincter. So before that, all of this can allow urine to move through the urethra. The external urethral sphincter has to be relaxed. That's a voluntary motor neuron. So that requires inhibition of our lower motor neuron. That comes down from the brain. Okay? To inhibit the voluntary neuron. So you may have been in a situation where you really wanted to go to the bathroom, but you couldn't. All right? So the home that I live in, I've shared this with you, I rent a bedroom from a good friend of mine from graduate school days. And it's her and her daughter and her granddaughter. They have 2 bedrooms upstairs. I have a bedroom downstairs, which is right off the family room, like about 5 feet. And the bathroom is a guest bathroom, but mostly mine, but when guests are there. So the daughter's boyfriend, the baby's daddy, comes over occasionally. Sometimes they're not getting along. Sometimes they are. One never knows. And I can hear people talking, water running, when I'm in my bedroom and they're in the kitchen 20 feet away. I can hear them talking softly in the family room. There's no way I'm going to go to the bathroom and tinkle while they're 10 feet away. It just doesn't work. I could go in there and I would sit there for 2 hours and nothing would happen. So I lie in bed and I wait sometimes till 2:30 in the morning for him to go home before I can go to the bathroom. Now, when I was a kid and we would drive 9 hours from Maryland to North Carolina, I would never go to any gas station bathroom. I would not go until we got to my grandparents' house 9 hours later. The worst it's been, a friend of mine from college who was teaching elementary school and high school, well, he needed me as a chaperone. They were going to go backpacking. We left at noon on Friday, got ready for bed up on the Cumberland Plateau in Tennessee, and I found a place in the woods. It would not work. Now, as a nursing student, I learned that you could help people go to the bathroom by listening to water. You turn on the faucet, take some warm water in like a ketchup bottle without the ketchup and dribble it on the perineal area. I've done my share of putting my hand in the sink and hot water. When you're outside, you don't have that, but I found a waterfall. Listening to the waterfall, nothing working. Get up the next morning, I've never had a trouble with that. Didn't happen. We're now going noon, 24 hours. What saved my life was a thunderstorm. Not the rainfall didn't help, but the camping trip got canceled. So Friday night, we're talking Friday noon to Saturday night at 11 o'clock, I got back to my apartment and my external urethral sphincter relaxed. So probably more information than you ever needed to know, but I'm very well aware of the fact that no matter how much you need to go, it doesn't work until the external urethral sphincter has been inhibited, the neuron has been inhibited, and that can relax, okay? Now, sometimes people after surgery, this system doesn't work very well. So would you give that person a cholinergic drug or an adrenergic drug? If they need to go to the bathroom after surgery and they can't go and they're facing the threat of a straight, quick in and out catheter before you allow them to go home on a in and out surgery. Would they be, so cholinergic is what? Sympathetic or parasympathetic? Parasympathetic. Adrenergic would be sympathetic. So which one's going to make, neither of them is going to make the external urethral sphincter relax, okay? But which one's going to make the detrusor contract? The cholinergic, okay? Side effect? Diarrhea or constipation? Parasympathetic cholinergic is a parasympathetic, so too much acetylcholine is more likely to cause the digestive system to work faster or to work too slow? Faster. Okay? So while trying to get the bladder to work, you might also activate the digestive system and cause diarrhea. So somebody who is incontinent, all right, and they don't want to go to the bathroom as much, are sometimes given drugs that are sympathomimetic. Okay? So you see those advertisements on TV. It's always a guy or guys riding their bicycles in the rain or canoeing in the rain. It's always water around them. Now they have a woman dragging her bladder with her through the bowling alley or from a speech or whatever. So that's too active a bladder. Okay? They're always feeling like they have to go to the bathroom. So those drugs are more likely to be adrenergic, sympathomimetic, causing the bladder to relax and not be so irritable. Okay? You want to keep the feeling from having to go to the bathroom. Suppress the urge. So that would be a sympathetic type of drug that would suppress the urge. All righty, let's take some questions or answer some questions. So which of the following, A or B, would result in a lower number of molecules of a particular substance in the peritubular blood than what's present in filtration? So let's visualize that. We have blood coming in the afferent arteriole into the glomerulus and getting filtered. So we have particles A in the filtrate. We didn't filter all of it, all right? So particle A is still going to be found in the efferent arteriole and then flowing into the peritubular capillary. So we have filtered particle A in the proximal convoluted tubule of the filtrate, and we have particle A in the peritubular blood surrounding the proximal convoluted tubule. So, would reabsorption of particle A or secretion of particle A end up in fewer of those particles than we started out before filtration? Okay. Now I erase this diagram? You guys have it? Okay. So let's draw a picture. Particle A. So what particle A gets filtered? The rest of it flows through the peritubular blood. All right? We have particle A here. So, reabsorption or secretion of these is going to end up with fewer of these particles in the peritubular blood. Does that help? Yeah. Okay. So which of these causes the peritubular blood to gain back particles, and which of these causes the peritubular blood to add even more to the filtrate? Looks like a wide-mouthed fanged snake there. So these particles were filtered. Okay. So are we going to have fewer particles in the blood if we do this, or if we do that? As the blood leaves, will we have fewer particles if we do this or that by the time the blood is down here? Okay. I just really, what I'm after is, do you know what reabsorption versus secretion means? Which way are the particles moving? Into or out of the filtrate? Okay? Question 2. Filtrate, which is entering the distal convoluted tubule. So we're looking at the function now of the nephron loop without mentioning the word nephron loop, except that I just did. So filtrate, which is entering the distal tubule, will be what relationship osmotically the filtrate that was in the proximal convoluted tubule. Since I'm in the realm here of drawing pictures.
I gotta check.
So the filtrate is doing this, and then this. So filtrate is in the distal convoluted tubule will be what relationship to filtrate here? What will be the, will this have more, or the same as, or fewer particles than it did as it entered the nephron? So does the filtrate go into the collecting duct as a hypo, a hyper, or an isoosmotic solution? You remember what it started out as? Starts out as 300 milliosmoles, and the surrounding solution is also 300. So what happens when it gets over here? Okay.
The number of particles goes up, and then it decreases. So this is hypo.
Would agree.
Okay? All right, one more question before we get to acid base. So we talked about at the very end of class on Monday, we talked about the adjustment of using hormones to adjust the particle component. So it kind of helps you with the answer to the previous question if you read it carefully. So what allows us to increase the concentration of the filtrate as it leaves the papillary ducts and goes into the minor calyx? What hormone do we use in the collecting duct? Now we use two hormones up there to concentrate the urine, but what hormone specifically in the collecting duct allows the osmotic concentration of the filtrate to increase? And I'll give you a hint. Think of the word aquaporin. What hormone causes aquaporins to be added into the cells, the principal cells of the collecting duct?
Yeah, I have it down as water reabsorption.
Yeah. The substances that, so it would make sense to get the osmolality up.
And again,
Yes.
that sentence should help you with the previous question. Okay, the information I provided.
Can you open water count? I'll go back to ADH rate. Can you open water count?
Exactly. That's why football players get in trouble for testing for ADH, because they're trying to pee out something that they're not supposed to be taking, and then they get popped on the drug test.
Okay, anyone need more time on this question?
Didn't know that.
right, so we're ready to look at acid base.
Maybe.
So here we have two beakers. I think this has 7 molecules of hydrochloric acid and this has 6, which is nice if they draw the same number. Hydrochloric acid and carbonic acid. We talked about this a little bit with the respiratory system. Hydrochloric acid dissociates in solution to release how many hydrogen ions? 1. It only has 1, right? Okay. Carbonic acid dissociates in solution to release how many hydrogen ions? 1.
Carbonate.
Okay. So, how does this affect pH? pH is a measurement of the concentration of hydrogen ions, all right? So if the concentration of hydrogen ions increases, does the pH become lower or higher? Lower. And a low pH is considered to be more acidic. As the concentration of hydrogen ions decreases, all right, the pH rises and is considered to become more basic or more alkaline. So it's a little bit backwards when you think of the pH versus the concentration, but you need to keep that in mind as we work with pH balance, all right? So hydrochloric acid is considered to be a strong acid. Carbonic acid is considered to be a weak acid. And yet we just looked at the fact that both of them release a single hydrogen molecule. We get 1 hydrogen ion. We get 1 hydrogen ion from every molecule of hydrochloric acid. We get 1 hydrogen ion from every molecule of carbonic acid. So that should help you pick out the correct answer here. Hydrochloric acid in solution dissociates 100%. So if you have 100 molecules of hydrochloric acid, you will end up with 100 hydrogen ions. If you have 100 molecules of carbonic acid, and I had a difficult time finding an exact number, but carbonic acid is a weak acid, and typically weak acids, about 10% of the molecules will release a hydrogen ion, okay? So from 100 molecules of hydrochloric acid, we have 100 hydrogen ions. From 100 molecules of carbonic acid, we have 10. Hydrochloric acid is a strong acid. Carbonic acid is a weak acid. Okay? So if we go back to this diagram, you can see that every hydrogen and chloride ion are separate, whereas we still have, this shows about half of the carbonic acid molecules are still attached to their hydrogen. When hydrogen is not an ion, it's not going to affect pH, okay? It has to be removed from or added to a particular solution to affect the pH. So if we add hydrogen ions to a solution, what's going to happen to the pH? It's going to go down. If we remove hydrogen ions from a solution, pH is going to go up, all right? So as you can see here, hydrochloric acid is never going to accept a hydrogen. It completely dissociates. So weak acids and weak bases, before I say too much, let's look at the next question. So this is actually 5. Whoops, that was the previous one, right?
Yep.
I want to go to a different one.
A, B.
A long time ago, this, and I think,
So a strong acid and a strong base, to give you the answer to this question, do not function as a buffer, because they're only driven one direction. They're either gonna completely dissociate or 99% dissociate and make the solution more acidic by releasing hydrogen ions, or sodium hydroxide, they're gonna remove hydrogen ions from the group, all right? So our buffer has to be one or the other. So how do we define a buffer then? A buffer is a molecule that can do either. It can either accept a hydrogen ion or it can give up a hydrogen ion, okay? A strong acid or strong base only does one or the other. So either a weak acid or a weak base functions as a buffer. So a buffer will either accept hydrogen ions So is it going to accept hydrogen ions under acidic conditions or under basic or alkaline conditions?
acidic conditions, right?
Acidic, because that's when we have too many, right? So when we're trying to buffer an acidic condition, we're trying to make the pH higher. And to do so, we need to remove hydrogen ions from the solution. So in that situation, a buffer will accept the hydrogen ion. Or it will donate or release hydrogen ions. So if the pH is too high, 7.6 instead of 7.4, then a buffer will release hydrogen ions under basic conditions or alkaline. Okay. What major organic type of molecule, lipids, proteins, or carbohydrates, is most significantly affected by the number of hydrogen ions? Why is buffering the pH of the body so important? Anybody have an idea? Proteins. Their shape will change depending on whether hydrogens are added or hydrogens are removed. So the pH is very critical to the function of enzymes. They might not even, so in our stomach, we have a pH of 1 to 2. The enzymes within the stomach function at that pH, but then the stomach contents move into the small intestine, and the enzymes from our pancreas, and those that are made by the small intestine, need a pH of 8 or 9. So we have to dump in a bunch of bicarbonate ions from the pancreas to buffer that pH, or we would never end up finishing digestion of our food. Right? So receptors, immunoglobulins, cell markers, pH changes, they have a different shape. Okay? One of the reasons why hemoglobin doesn't hold on to oxygen when the pH is lower. Okay? It's changed its shape and it doesn't fit in to bind with the iron molecule very well.